Hey, good morning, everyone. I'm Luan I'm a mathematician at Harvard Medical Foundation, and I'm going to talk about version 2 of CUPPA, which is a tissue of origin classifier. So why do we want to classify tissue of origin? So let's say we have a patient that has a metastatic breast tumor, and if we would know that it came from the lung, then we could treat it with lung cancer-specific drugs. But how do we know that the metastasis is from lung? Well, usually this is done by histopathology, but this is not successful all the time. And in these cases, these tumors are referred to as cancers of unknown primary. So basically, we don't know the tissue of origin. And because of that, we cannot, well, these tumors do not have, these patients do not have many treatment options. So in these cases, we can use whole genome and transcriptome sequencing to resolve these tumors. And so what kind of features can we use? So firstly, we can use DNA or mutation features. So the first type is we can look at the genomic distribution of SNVs. And this kind of is a proxy for the epigenetic state of the tumor cells. Well, we can also look at the trinucleotide context, and this kind of reflects the mutational processes that have been active in the cell. So for example, UV radiation in skin cancer. And lastly, we can look at other mutation features such as driver mutations, gene fusions, and viral insertions. Besides DNA, we can also look at RNA. So the obvious one is gene expression, but we can also look at fragments with alternative splice junctions, and this is basically the expression of everything else other than genes. So at Hartwig, we use an in-house pipeline that analyzes DNA and RNA sequencing data, and this pipeline produces all of the input that CUPPA requires. And at the very end of this pipeline is, well, at the moment, CUPPA version 1, let's say previous version of CUPPA. And this version of CUPPA is a rule-based classifier, but in version 2, we've moved to a machine learning-based classifier. So version 2 is essentially layers of logistic regressions. And so in this first layer, the 5 feature types that I mentioned previously flow into their own logistic regressions, and each logistic regression produces a bunch of cancer type probabilities. And these probabilities are then aggregated by another layer, which produces DNA combined and RNA combined probabilities. And the last layer multiplies these probabilities together, and then you have an overall probability. So this model was trained on a large pan-cancer data set of about 7,000 tumors. This data set is composed of samples from Hartwig itself, but also from the PCOG consortium. This data set has 40 cancer types, and you can see in the blue and orange columns the samples that have, or the number of samples that have DNA only, but also DNA plus RNA. So how does CUPPA perform? So if we take a look at only the combined classifier, which uses both DNA and RNA, we expect to predict about 90% of samples correctly. But in the cases where we don't have RNA, we can only use DNA, then we expect to predict 88% of samples correctly. In a clinical setting, we actually don't use, we have a probability cutoff of 0.8. So we only accept high confidence probabilities or high confidence predictions. And in those cases, we expect to predict about 95% of samples correctly. So in comparison to version 1, CUPPA version 2 gets about 1 to 3% increase in recall in the fraction of samples correctly predicted. But more importantly, if we look at the number of samples that get a high confidence prediction, then in version 2, we get about 6% more samples. And this is really important because then these samples, these CUP samples or patients actually get treatment. And if we look at the misclassifications, then we actually find that many of the misclassifications are to cancer types that are of the same supertype. So in this confusion matrix, the diagonal shows the number of correct predictions, and the off diagonal shows the incorrect predictions to other cancer types. And you can see that, for example, in breast here, most of the samples are predicted as either triple negative or other breast cancers, and similarly for other cancer types as well. So the nice thing about CUPPA is that it's not a black box. So each prediction gets a visual report. And so this is a real sample that has been predicted as non-small cell lung cancer. But in addition to the probabilities, the visualization also shows why these probabilities came about. So for example, in this sample, there's a smoking signature, which is, of course, characteristic of lung cancer. This sample also has an RB1 driver mutation, which is specific to small cell lung cancer. And we can also see that small cell lung cancer is a high-performing cancer type based on training set cross-validation. So based on this information, we can conclude that we can trust that it's actually a small cell lung cancer. Okay, so doing the same exercise for the 350-odd samples in the Hartwig dataset without a known cancer type, we can actually resolve many of them. So on the left here, you see the high confidence samples, and many of them have known features. So for example, we have some samples from some anogenital and head and neck samples with human papillomavirus insertions. We have a bunch of some additional lung cancer samples with the smoking signature and some known driver mutations. Tempus-ERG fusions in some prostate cancer samples, and UV radiation signature in some skin cancer samples. There are also a bunch of less confident samples, so samples with a prediction of 0.6 to 0.8, which we can also resolve. And of course, there are about 30% that remain unresolved, but about 60 to 70% can be resolved. Okay, so how can we run CUPPA? So I expect version 2 to be in the Hartwig pipeline by the end of this year. The Hartwig pipeline itself can be run from a Nextflow implementation. And the data set that I used to train the model is publicly available as well. And the links you can follow through this QR code. And I'd just like to acknowledge all the people that have worked on this project, especially Peter and Charles, who were part of the core development, and I'd be happy to take any questions.
Great. Thank you, Luan, for the wonderful talk. Do we have any questions for Luan?
Thank you so much. For individual logistic regression, I wonder how you have done feature selection across RNAs and DNAs.
So for feature selection, I've used the chi-square test, and that's kind of a multi-class feature selection method, and that was done for the driver mutations as well as for the gene expression and alternative splice junctions.
Great. And next question.
Thank you so much. Given the rare situation of some cancer type, how your classifier handle imbalanced data set where you have some tissue type underrepresented?
So for the underrepresented cancer types, the model just doesn't perform well. So if we go back to this slide, in the bottom, you can see that there are some cancer types with yellow in the heat map, and they just don't perform well. But we do report this in the visualization so the doctor can know not to trust these cancer types as much.
So for the model prediction results, you saw here is the micro average accuracy or the balance accuracy?
This is
Because-
micro average.
Okay,
Yeah.
because it seems to me that you have, for some cancer type, you have so many samples, and some others you don't have that, right? So maybe your model performance, I mean, focus only on the samples that they have, only cancer types that have a lot of sample.
Yeah, for sure, the cancer types with more data do perform better, as you can see in the bottom heat map as well. I do counteract this by doing class balancing in the logistic regression, but of course that doesn't completely solve the issue. Yeah, the only way to solve this issue is to get more data.
Okay, I have another question.
Yeah.
Have you ever considered to add sex into your model?
At what, sorry?
Sex and gender.
Sites?
Yeah, gender, male and female. Because it seems
oh.
to me, yeah, right. Because like breast cancer, it never happened in the male, and prostate cancer can never happen in female.
Yeah, this is a feature in the model.
Okay.
I think you can see it. Yeah, it's the first one. It's the first row on the second heat map. There's a feature called is male, so the feature is in there.